Welcome into the lab. So today we're going to explore what is arguably one of the most mind-blowing mysteries in all of biology. We are looking at an epic construction project, one where a single fertilized egg cell basically acts as the ultimate master contractor. I mean, it's managing countless moving parts just to build a complex, intricately detailed animal body from scratch. It is this absolutely incredible journey from microscopic simplicity to staggering macroscopic complexity.
Take a second to really think about this question. How did a single cell develop into this intricately detailed embryo? As a developmental biologist, I'll be honest, I never stop marveling at the sheer elegance of life. It's a literal biological miracle that happens every single day, right all around us, and yet it still remains one of the most complex, perfectly orchestrated architectural feats in the known universe.
Okay, let's dive into this. Here is our syllabus for today's construction project. 1. Our biological blueprints. 2. Cleavage, the first divisions. 3. Gastrulation and germ layers. 4. Comparing embryonic construction. And 5. Human implantation and the placenta.
Section 1. Our biological blueprints. Before we tour the actual cellular work site and, watch the walls go up, we need to meet our biological contractors. You might be wondering why biologists don't just study human cells to understand human development. Well, we rely heavily on model organisms in the lab. We're talking sea urchins, frogs, chicks, and nematodes. Why? Mainly because they have incredibly short life cycles, and we can easily observe their development. But the absolute most crucial reason is that animals share a common set of regulatory genes. Actually, get this, a gene that specifies eye formation in a mouse can direct eye formation if you experimentally introduce it into a fruit fly. Studying these animals reveals this universal micro to macro evolutionary connection that governs how all animal bodies are built.
Section 2. Cleavage. The first divisions.
Once fertilization happens, the worksite kicks into high gear. The pacing becomes incredibly rapid as we start this non-stop cutting of our raw biological materials. We call this initial high-speed phase cleavage. And here's a really fascinating distinction. During cleavage, the embryo doesn't actually grow in mass at all. It essentially skips the growth phases of a normal cell cycle. It's merely dividing its existing cytoplasm over and over again, rapidly carving itself up into smaller and smaller cells called blastomeres. Eventually, it forms a hollow ball of cells known as a blastula.
Now, what's really interesting here is how stored nutrients, the yolk, basically force the embryo to use different construction strategies. Simple organisms like sea urchins have very little yolk, so they just divide evenly. In frogs, an asymmetric yolk kind of slows down cell division at the bottom, but the cut still passes completely through the egg. That's a process called holoblastic cleavage. But contrast that with myrtoblastic, or partial cleavage in chicks. A chick egg has such a massive, dense volume of yolk that the cleavage furrows can't pass through it at all, so division is restricted entirely to this tiny whitish layer sitting right on top of the yolk.
Section 3. Gastrulation and germ layers.
After all those rapid-fire divisions of cleavage, the pace finally slows down. The embryo enters this highly deliberate, orchestrated phase called gastrulation. This is where that hollow blueprint completely reorganizes into a three-dimensional structure. It is an absolutely incredible feat of biological origami. The embryo takes that single sheet of cells on the surface of the blastula and pushes groups of them inward, establishing the ultimate architectural layout. Cells change positions, they get new neighbors, and they set up the deeply layered foundation of the entire animal body.
This biological origami results in 3 foundational materials, which we call the embryonic germ layers. On the outside, we have the ectoderm, which forms your skin and your nervous system. In the middle is the mesoderm, which gives rise to your muscles, your skeleton, and your circulatory system. And on the inside, the endoderm forms the lining of your digestive and respiratory tracts. Every single complex organ you possess, your whole body, originates from one of these 3 specific distinct layers.
Section 4. Comparing embryonic construction.
So we know what foundation materials need to be laid down, right? But as architects of life, it is fascinating to look at how different organisms take totally different approaches to get the exact same inward cellular migration. A sea urchin, for example, just pushes its outer layer straight inward, kind of like poking a deflated ball. But the frog embryo, it uses a circular folding crease called the blastopore lip, rolling a sheet of cells inward over that edge. Meanwhile, the chick embryo, which is developing flat on top of that massive yolk we talked about, uses a midline pileup of cells called the primitive streak. Cells detach and migrate downward and sideways through this central crease. Different blueprints, but the exact same magnificent outcome. But how do the cells actually perform these movements? I mean, what's doing the heavy lifting? That brings us to morphogenesis. The actual physical machinery doing the work here consists of your cell's cytoskeleton.
Microtubules act like these scaffolding cranes to elongate the cells, while actin microfilaments act like tightening pulleys to constrict one end. This wedges the cells into new shapes and drives processes like convergent extension, where a whole sheet of cells narrows and lengthens to wedge the embryo into its new form. Literally, microscopic cranes and pulleys.
Section 5. Human implantation and placenta.
Let's move to our own species and see how this builds human beings, transitioning from our model organisms to mammalian development. In humans and other mammals, our version of the blastula is called the blastocyst. It's a sphere of over 100 cells, but I really want you to clearly grasp one uniquely mammalian term here, the inner cell mass. Clustered at one end of the blastocyst cavity, this specific group of cells is exactly what will develop into the actual human body. The outer layer, well, that has a completely different, highly invasive job.
Trace this miraculous early timeline with me to see just how quickly a human embryo sets up its initial construction site. By day 6, the blastocyst reaches the uterus. Day 7, it implants into the uterine lining. By days 10 to 11, extra embryonic membranes start to form. And by day 13, gastrulation officially begins. Think about that. In less than 2 weeks, the work site is fully secured and complex bodybuilding is already underway. Let's walk step by step through that dramatically invasive process of implantation. When the blastocyst reaches the uterus, that outer layer of cells, which is called the trophoblast, aggressively secretes enzymes that break down the uterine lining. It literally invades the endometrium, capturing maternal blood vessels to secure the site. From there, the extra embryonic membranes form around the embryo and the placenta takes shape.
And this brilliantly illustrates the ultimate engineering marvel of mammalian development, the placenta. Because we are terrestrial amniotes, we require a highly specialized system to actually thrive inside a fluid environment within the mother. The placenta acts as this unparalleled life support logistics hub. It is constantly exchanging vital nutrients, oxygen, and nitrogenous wastes between the developing embryo and the mother, making our entire complex internal development possible.
So we've gone from a single dividing cell to a fully implanted multi-layered embryo ready to build complex organs. Exploring this epic construction project really just leaves us in complete awe of developmental biology, and it sparks a pretty provocative question to end on today. If a single cell holds the blueprint to build an entire human body from scratch, what profound secrets are adult cells still hiding? Keep questioning, keep exploring, and thank you so much for joining me in the lab today.